All right, so thank you everyone for joining. I am Mohammed Abdul Halim. I usually go by Halim. I am a postdoctoral fellow working under the supervision of Professor Dr. Sean Thomas at the Institute of Forestry and Conservation, Daniels at the University of Toronto. Today, I'll be talking about greenhouse gas flux patterns from green roof substrates and some surprising effects of biochar, mainly from our Grid Lab 2 experiment. So let's get into the details. All right, so green roofs uniquely combine people, building, and environment, and a sustainable way to make our cities climate smart. And they offer several ecosystem services. Those are critical, but otherwise lost from rapid urbanization. The carbon sequestration potential of green roofs is of such a recent interest as rapid urbanization is disproportionately responsible for ecosystem carbon loss. And appropriate management of green roofs can potentially compensate for carbon loss from urbanization. So understanding the carbon fluxes from green roof systems are very important to design green roofs that can actually sink carbon. So a little bit of mechanism. So green roofs exchange carbon with the atmosphere primarily via plants and substrate, and in the form of carbon dioxide and methane. And the net effect of carbon dioxide and methane, efflux and influx, will determine when or whether a green roof is a sink or source of carbon. In this study, we're mainly interested in CO2 and methane flux from the substrate, not from the plant leaves, because most of the methane exchange happens at the substrate level. So it is therefore important that we understand the magnitude and the direction of carbon dioxide and methane fluxes, their drivers, and the ways we can enhance them to help design green roofs that actually sinks carbon. Our knowledge from temperate forests suggests that substrate temperature, moisture content, pH, electrical conductivity, organic matter content, species composition are some of the key factors that might affect the carbon dioxide and methane fluxes from green roof. Our better understanding of the relationship between the direction and magnitude of carbon dioxide and methane fluxes with their drivers will help us to make design-specific decisions for green roofs. So how much do we know about the magnitude and drivers of substrate carbon dioxide and methane fluxes from green roofs? Among various ecosystem services, carbon sequestration potential of green roofs are, as I mentioned, of recent interest. However, our understanding of the green roof's role in carbon sequestration is limited. Some previous studies looked at the carbon sequestration potential of green roofs, and they found that green roofs has the potential to sink carbon, and they, however, completely overlooked methane fluxes, a 30-time more potent greenhouse gas compared to carbon dioxide in terms of the global warming potential. And there is only one study that we are aware of, is Tim-Suk et al. 2019 study published in the Science of the Total Environment journal. They quantified greenhouse gas fluxes using a closed static chamber method for 3 representative green roofs. And they found higher ecosystem respiration in a deeper sod roof system than in a shallower or non-substrate. And also they observed net methane uptake in all systems. We are not aware of any study that has quantified greenhouse gas fluxes in a green roof system based on replicated measurements as opposed to measurements of representative systems. Moreover, the Tim-Suk et al. Study, they used the static chamber technique, which inherently suffers from several limitations and tend to underestimate fluxes. So to overcome some of the limitations discussed and to further our understanding, we set up an experiment at Grid Lab 2, Spadina 1, experimental extensive green loop at the University of Toronto. Using the dynamic closed chamber technique in replicated green loops, we looked at the magnitude and drivers of substrate carbon dioxide and methane fluxes from green loop substrate. We're also looking at the effects of Biochar on substrate carbon dioxide and methane fluxes in the green loop. So the question is why we're looking, where we're looking at the Biochar treatment, as we have already heard a lot of fantastic discussion on the properties of Biochar as an amendment to soil. Biochar are also known to reduce carbon mineralization. They enhance water retention capacity, and even more in some system, they can increase methane oxidation. So to test how Biochar actually performs, we surface incorporated sugar maple Biochar at a 20 ton per hectare dose in 12 beds and left another 12 beds as control. We have one species sedum throughout this 24 bed. This is part of the experiment, just to be clear, because we have a native plants, but at the time we're doing this measurement, they did not have sufficient cover to compare with the sedum, so that's why we're partially presenting the data here. So our results from the Grid Lab 2 experiment indicate that carbon dioxide flux ranged from 1.3 to 5.1 micromole per meter square per second, and methane fluxes ranged from minus 3.5 to 1.14 nanomole per meter square per second. And some of the highest methane influx that we observed are comparable to the one that is measured from nearby urban forests. This suggests that green loops could potentially be designed to act as methane sinks of comparable importance to urban forests, maybe possibly by using Biochar. So let's see how Biochar is helping or doing with the carbon dioxide and methane fluxes. Our results indicate that Biochar had no significant effect on carbon dioxide fluxes. However, they significantly improved the methane consumption. In the control plots, the methane consumption rate is similar to the one that we observed at Grid Lab 1 in our another experiment. But in Biochar plot, the consumption rate is about 200% higher than the control plots. So the magnitude of both CO2 and methane fluxes varied with months of measurement. But for the methane consumption in Biochar plots, throughout the measurement months, we observed higher methane oxidation. So this is the measurements from our 2020 field campaign. We have done one measurements in 2021. We have not finished processing the data yet, so I'm not able to present that yet. So in terms of the drivers, we found that CO2 fluxes were generally driven by substrate moisture content, temperature, pHs, and the interaction of electrical conductivity, moisture content, and pHs. On the other hand, methane fluxes were primarily driven by substrate temperature. So Biochar seems to be driving the methane consumption pattern by affecting substrate temperature. In summary, the observed CO2 flux from Grid Lab 2 suggests green roof substrates are unlikely to be an appreciable source of atmospheric carbon dioxide as a direct result of aerobic decomposition processes. And extensive green roofs are a weak sink of methane, but a Biochar addition can significantly enhance the methane consumption rate, making green roofs comparable to the methane consumption rates observed from urban forests. So overall, our data supports that the methane sink could be another environmental benefit of Biochar incorporated green roof system. So that's all for my talk. Thank you for your attention. If you have any questions, please let me know. I'll try my best to answer.